In this session, we're going to introduce a third kind of subspace clustering methods called bi-clustering methods. What is bi-clustering? Bi-clustering is to cluster both objects and attributes simultaneously. That means we treat objects and attributes in a symmetric way. Let's look at an example. Suppose we got a gene expression data analysis. We have rows. Every row represents a gene. We have m columns. Every column represents a sample or a condition. Essentially, these are the real value represented to the expression level. Then we have 4 requirements to find biclusters. The first requirement is only a small set of objects participate in a cluster. That means only some genes participate in a cluster. Then the second requirement is a cluster only involves a small number of attributes. Simply says only some columns are involved in the cluster. Then the third one is an object may participate in a multiple clusters or does not participate in any cluster at all. Simply says some genes, some rows, some genes may be in multiple clusters, but there could be many genes that are not in any cluster at all. The fourth one is an attribute may be involved in multiple clusters or is not involved in any cluster at all. That means some columns, like this condition, this sample, could be involved in more than one cluster, but there could be many columns. They are not involved in any cluster at all. Then a typical example is a gene expression or microarray data. What you got is this matrix we call gene sample or gene condition matrix. So each element in the matrix represents real number. It records the expression level of the gene under certain condition. Another example could be customer and product, their relationships. This simply says one customer may be involved in the bicluster in more than one cluster, but some product also may be involved in more than one cluster, or some product may not be involved in any cluster at all. Now we look at the types of biclusters. Suppose we get a set of genes and a set of conditions. The bicluster essentially tries to find a submatrix like this. The genes and conditions follow some nice consistent patterns. We can look at different ideal cases. Could be we may find one kind of bicluster could be every value just like a constant. Like if this one is 10, this are all 10s. This is an ideal constant value subcluster. And this bicluster is very ideal, but barely you can see such cases. The second case is the bicluster with constant value in rows. That means each row in this bicluster, they have the constant value. Okay. The third one could be the bicluster have some coherent value. That means they have certain pattern. This AKA means as known as pattern-based clusters. Okay. For example, you can see here, you may observe some relation like if you increase by 10, so everything just like a copy. And here you increase 30, every row like a copy. You decrease by 50, every row also look like a copy. That's also ideal case. The last one could be if all these rows and columns, they have the relationship, it's hard to find. For example, this increased by 10, this is by 30, but this is by 50, this is by 0. Okay. If you find this, what is the relationship? You may find relationship is the increase or decrease follow the same trend. Simply says you look at the corresponding 2 cells, you will say if they increase, the other one is also increase, their product will be greater than 0. Okay. Or one increase, the other one at least maintain the same, so it's equal to 0. If they all decrease, that simply says you get negative and negative, they multiply is still positive. So that means you're only interested in up or down regulated changes. In that case, this is another kind of pattern. Okay. Then in the real world data, they cannot be so ideal like we illustrated above. That means the real world data is noisy. We try to find not ideal biclusters, but we try to find approximate biclusters. Essentially, there are two kinds of methods. One called optimization-based methods, another called enumeration-based methods. The optimization-based method is trying to find submatrices only one at a time, but we try to achieve the best significance as a bicluster. Simply says we try to find the best one we can find at each time. However, the computation is very costly. Try to find best one may not be quite realistic. The best is to use greedy search to find the local optimal biclusters. That means locally this is best, globally it may not be. Okay. Then one such algorithm called delta bicluster algorithm developed by Chen and Church in ISMB, published in the International Symposium of Molecular Biology, Bioinformatics Conference. Another one is enumeration-based method. The method essentially to use a tolerance threshold. That means as long as they are approximate, they follow within certain threshold, we can specify the degree of noise allowed in the bicluster. Then we try to enumerate all these submatrices as biclusters that satisfy the requirements. Try to find this is like a pattern analysis. So one algorithm called delta P cluster algorithm developed by Haixun Wang's group, published in 2002 Sigma conference. Their further refinement called MAPLE was published by Jian Pei and others in ICDM conference.